This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 1225, recorded on June 6, 2025. There's something symmetrical there. 1225, 2025. Not really, but I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody. Here it's 69 Fahrenheit, which is 23 Celsius, and I don't really see any blue sky in between the clouds, but we have some of that bad air coming down from the wildfires in Canada, so it's just kind of hazy.
Here it is 29 Celsius and mostly sunny. It's a very nice day today. Well, this is the last 2 days, and it feels like summer, finally. Not that it matters, but, you know, the other days didn't feel like summer. They had that spring twinge in them. But today and yesterday feels like summer. And I'm still wearing thermal turtlenecks and wool jackets because it's cold here at the incubator, and I can't regulate temperature. Yeah. Also joining us from Western Massachusetts, Alan Dove.
Good to be here. Maybe you should call the incubator the minus 20 or something.
Yeah, the fridge.
It's here in Western Mass. It's actually kind of hot and muggy. It's 86 Fahrenheit, 30 C, cloudy, and we are expecting thunderstorms, possibly severe, and there's a flood watch in effect for tonight.
Weather, you can't do anything about it, Alan.
Nope.
You can
Also,
mix all that with our unhealthy air. I forgot to say that. It's 159.
Wow.
Oof.
Also joining us from Austin, Texas, Rich Condit.
Hi, everybody. It's 96 degrees here, clear blue skies. We're looking at mid to high 90s for the next several days, and then dip down to the 80s for a while with some thunderstorms. That would be welcome. Here we are in Texas. It's Texas summertime.
Texas summertime, Texas measles.
Yeah, that too.
Well, good stuff. Speaking of good stuff, if you like our programming, now we're programming. If you like our programming, we give you science, real science, no lies, no misinformation. This is the place to get. There are other places you could get good science from too, but we're one of them. And so I'm going to tell you about us. We need your support to continue. Microbe.tv/contribute. We'd appreciate any amount you can spare. Dixon's birth anniversary was yesterday, June 5th. He was born in 1940, so that would have made him 85. Just thought I'd note that, and I think of Dixon all the time because I left his name on all the documents. And, he was with us since 2008 here on
From
TWiV.
the beginning?
From the beginning.
So I knew him for many years, and so yeah, I do miss him.
Hi, Dixon.
Hopefully he's podcasting somewhere else.
I'm sure he is. Telling stories.
Or fishing, right?
Fishing.
Yep.
Speaking of podcasting, what's up with ASV, Kathy?
Well, there will be a podcast there. TWiV is going to be presenting on Thursday of that week, and the meeting starts on Monday. July 14th to 17th are the dates at McGill University. Well, hosted by McGill. It'll be held at Montreal at a big convention center, the Palais de Congrès de Montreal. And so most of you have probably registered by now. If you haven't, you're paying the slightly higher price. But if you get going and you do it before you actually get to Montreal, you'll save money because in person it costs even a little bit more. So register by June 30th in order to get the best rates at this point. And we pointed out last week, but I'll just mention it again, the timing of the events has been changed relative to previous years. So the first day starts in the morning with satellites, and then right after lunch is the welcome reception, and then the keynote address, and then the first plenary session with, I guess, 4 or 5 speakers. I haven't counted. And then the first evening workshop with people presenting their talks about their own work. And so it's quite a bit different. And then, as Brianne pointed out last week, that means the last day is different too. The scientific sessions run through about dinner time, when there will be a big dinner and followed by a dance that everybody's invited to. So check out those details at asv.org/ASV 2025.
I was at the convention center once there was an International Congress of Virology there some years ago. I did a TWiV there. And I remember I came out, it's on a whole block, and I was waiting at the corner for the light, and these two cars smashed into each other. And one of them was like drifting towards me. And I'm like, what?
Whoa.
And I finally ran away. You feel weird running, but the car didn't get up on the curve, fortunately. But it just emphasizes how fragile everything is.
Yeah.
Things could happen. Like a few months ago, there was a derailment of a freight train on the rail line that I take, and it disrupted everything because the freight, and then now the freight and passenger lines share tracks, parallel tracks. So every time a freight train passes me, I think if that derails now, we're all toast. Because the derailment breaks all the tracks, the cars jackknife and they go all over the place. It's a mess. And they've left all the cars lying by the side of the tracks, all the broken wheels and the broken cars to remind you of how perilous our world is.
I have to say, my recent trip to Japan, there were a number of times when I was in certain places, I thought, if there were an earthquake right now, I would be toast.
Yeah.
Because they just didn't seem like they would hold up. They were old places, pre-earthquake standards. And
yeah.
Well, but for those of you thinking of taking the train to Montreal to go to ASV, don't worry. It'll probably be fine.
It'll probably be fine.
And you probably won't be next to a car crash outside the convention center either.
And there probably won't be earthquakes.
Right, but very unlikely that you get earthquakes there.
Yeah, so we're just preying on the rare events that we have encountered.
Yes.
Right? This is true.
Kathy,
for the most
aside from being the mouthpiece for ASV for TWiV, do you have anything to do with this anymore, or are you done?
I'm pretty much done.
Okay.
Yeah.
So you're going to go and enjoy the meeting, right?
I can. Yeah. Just go enjoy the science, talking to people, hearing new, cool virology.
Well, I, for one, appreciate your service over the years because that's a big job, and you did a good job.
Thank you very much for that. Okay, a couple of news items here. The first, I'm sure you've heard of these, but, we can do face plants on them. This is a Nature article by Jeff Tollefson and Dan Garisto. Trump's call for gold standard science has prompted an outcry. So maybe you heard this executive order signed last week, mandating the overhaul of research integrity policies to ensure that the federal government promotes gold standard science, more like fool's gold standard science, to ensure that science is transparent, rigorous, and impactful, which I already thought it was. Now they're making it less transparent, as we all know.
Well, what they're doing is they're empowering political appointees to jump in and embrace fringe theories and,
a political appointee can tell you can't do that research if they don't like it.
Yeah.
It's no longer science by committee. It's science by edict. So this is really bad. There are lots of quotes here from people saying why it's bad.
And of course, the administration is couching this in terms of, it'll be, transparent and objective, but it's going to be anything but that.
The funny thing is they blame Biden for misusing scientific evidence,
In other words, for citing scientific evidence they didn't like.
Yes, but we should remind everyone that the U.S. shut down in March under Trump's orders, okay?
Yeah.
So he's done it before. He's not the mouthpiece. Well, he's the mouthpiece, but he's not the brains behind all this stuff. It's that 2025 book.
So,
whoever wrote that.
Several things struck me about this. I'm always struck by, without going into too much detail, the irony of these sorts of orders and et cetera, claiming to reverse or undo the politicization of science, when it seems to me that's, it's the opposite. Though I'm sure it's a matter of perspective. But one of the things that in that regard that struck me was in this article, they talk about getting comment from a spokeswoman for the U.S. Office of Science Technology Policy, OSTP, who said, among other things, she accused the Biden administration of incorporating, quote, radical woke ideology into the scientific enterprise by introducing diversity, equity, and inclusion programs. And, that smells political to me, and it smells Project 2025 to me. The other thing I would recommend that people do is actually go read this executive order. In the article, it's linked in the first paragraph, and you can read it for yourself. And there's a couple of things that stand out to me. One is the process of restoring the gold standard science. Within 30 days of this order, the director of the Office of Science and Technology Policy shall, in consultation with the heads of relevant agencies, that's vague, the heads of relevant agencies, that's not specified, issue guidance for the agencies on implementation of the gold standard science. So the director of OSTP is going to tell the scientific community via these agencies how they should behave.
Yeah.
And then one of the things I noticed is that once those guidance is basically established, each agency head, as necessary and appropriate, and in consultation with the director of the Office of Management and Budget and the OSTP director. Director of the Office of Management and Budget is Russell Vogt, who's a major author of Project 2025, and that's where a lot of this bashing of the radical woke ideology comes from, straight out of Project 20. I'm not making anything up here.
No.
And then there's a section that details what needs to be done, called improving the use, interpretation, communication of scientific data. And if you read through this, it is mountains of paperwork to be done in a short period of time. So I'm not real optimistic about this whole thing.
No.
All right, thank you for that, Rich. Our other news item, I have an article from Stat by Anil Oza. Corrupt, in quotes, medical journals have to change, RFK says, or NIH will publish in-house. The health secretary said Lancet and New England Journal of Medicine and Journal of the American Medical Association have been influenced by the pharmaceutical industry. And I thought you might have some thoughts on this, Alan.
Yeah, so I saw, when I first saw headlines about this with, RFK Jr., criticizing medical journals and the NIH might publish in-house, I was like, oh, maybe they're actually doing something right here. Because, there are legitimate criticisms of subscription-based medical journals, and we've talked about this on TWiV, things that are paywalled, that were done with taxpayer money, and the open access movement for, 20 years has been working on addressing this issue and has done so imperfectly. But, I thought, well, gosh, this might actually not be a horrible idea. But no, his criticism is not about access to the journals. It's about claiming that the research is paid for by the pharmaceutical companies because they're advocating these dangerous vaccines and what have you, I'm sure. And so he's using that as the basis of his criticism, which is wrong. These journals actually have detailed conflict of interest policies that people have to disclose what their affiliations are, and so on and so forth. And a lot of the research being done at pharma companies, by the way, isn't even published in a timely manner because it's proprietary. So what's going out in these journals is the basic research, which of course has now been defunded. But, to the extent that the taxpayers paid for it, we ought to be able to see it, and it should be published someplace like this. And I'm not optimistic about the NIH publishing the work in-house either, since they've been defunded. I don't know who's going to actually do that publishing work. So this whole thing is just wrong-headed.
So he says the journals are all corrupt. So no. I'd like to know what the evidence for that is.
No, they're not corrupt. They're the for-profit ones are certainly for-profit, and I have my own criticisms of some of the publishers who take that a little to the extremes and leverage their monopolies. But that's a totally different subject from this.
Yeah.
It's like it doesn't even address peer review.
No. Yeah.
Well, he makes up his own problems. The real problems he doesn't know about because he doesn't know anything about science.
Or he doesn't want to hear about.
Maybe that too. I prefer to think he doesn't know about it, but you're right. He doesn't want to hear it either. Every day there's something new, and that's just two of them. Okay, so on to science. Science is still being done.
In China, anyway.
In China, as you will see, both papers out of China. Kathy, do you have a summary of the snippet?
I do.
I can do authors and affiliations if you.
Oh, I'm ready with that. Actually,
are you ready with that?
Yeah.
Too?
So,
okay.
Alan, you have to do the paper summary because Kathy didn't do that. Okay.
For the second one.
Yeah. So I just saw this in the notes that the main paper you didn't do a summary. I didn't either. So we'll improv that one.
Okay, we'll improv it.
Or Rich will do it.
No, you can improv it.
Okay. Anyway, the title of the paper is Genetic Diversity of H9N2 Avian Influenza Viruses in Poultry Across China and Implications for Zoonotic Transmission. So there are 7 co-first authors, Jing Yang, Joan Li, Zhu Sun, Jiangming Li, Guang Hua Fu, Chen Chan, and Yangchun Yang. And there are 21 other authors, including 3 that you can correspond with, who I'm taking to be the senior authors, Weifeng Shi, George Gao, and Yuhai Bi. And they come from a variety of places. The first 3 are from Beijing, the Chinese Academy of Sciences, and the Research Center for Respiratory Infectious Diseases. And then one from Jinan, which is the Shandong First Medical University and Academy of Medical Sciences. And then College of Veterinary Medicine at Jinzhang in China, Shangji Agricultural University. One from Fuzhou at Fujian Academy of Agricultural Sciences. And one from, Hangzhou, the couple places, including College of Veterinary Medicine of Zhejiang A&F University and College of Animal Science and Technology. And then 8 other affiliations. So it's a large crowd of Chinese people doing this research. And so what are they doing here? Well, surveillance of influenza in birds is important, and China has been doing it actively and on a monthly basis since 2014. So here is sort of the latest update in their report on surveillance. This is for the 4 years from 2019 to 2023. They sampled over 18,000 poultry samples and found that about 1 in 6, or 17.6%, were positive for avian influenza. And they found that one subtype was dominant. That's the H9N2. And just to remind you, for influenza, H is for the hemagglutinin surface protein, and N is for the neuraminidase surface protein. So this is H9N2. And this subtype was also predominant in their previous reporting period from 2016 to 2019. And in the period before that, from 2014 to 2016, H9N2 was dominant in northern China, and another type, H5N6, was predominant in southern China. So there's some change over time and over geography. So they want to understand the prevalence over time so that they can evaluate the risks to both poultry and to people, the risk of zoonosis of these viruses crossing out of the avian reservoir into birds and potentially other mammals, as we've seen recently with avian influenza H5N1 in cows. So they developed a new phylogeny or evolution-based naming system so that they could classify what's going on over time, and predominantly in this H9N2 group. And in that group, they found 10 subclades of hemagglutinin of this dominant H9N2 lineage. And not surprisingly, they found antigenic variation in these subclades. And then they kind of drilled down a little bit and found that 99% of these had an amino acid change in the hemagglutinin, and that is involved in receptor binding. So keep that in mind. 96% had an amino acid change in the NP protein, which is one of the subunits of the viral polymerase, so having to do with replication. But in this case, that particular change has something to do with resistance to the host immune response. So now the most predominant viruses have this change associated with receptor binding and a change associated with evasion of the immune response. And at the very end of the paper, they talk about 3 factors that are important for zoonosis that we need to be concerned about. And those are 2 of the 3. And so then they go on further to say that viruses that have these 2 amino acid changes and a third one, which is in another gene of the replicase complex known as PB2, a virus that had all 3 of those viruses were transmitted to naive guinea pigs and ferrets, both through direct contact and through respiratory droplets. So the implication is, if they could go from this avian virus to these 2 mammalian models, that perhaps it's something we need to be concerned about. And so these viruses are interested from the standpoint of zoonotic potential and potentially crossing to humans. And I leave it to the rest of the guys here to go into any details that they want to.
I want to probe a couple of other little background pieces of information. First of all, how many hemagglutinin types or subtypes are there? It's like 19. Is it that many?
18.
18 is what I remember. Yeah.
Okay.
And 11 NAs.
Okay. And so of those, the different, I mean, we're talking about subtypes and subtypes here and antigenic variation within subtypes. But one of the things I think to keep in mind is that the different primary subtypes, HA1, HA2, HA3, and et cetera, are immunologically quite different, right? Fair enough?
Yeah.
And of those 18, they all circulate in birds, and only 3 of them have circulated broadly in the human population. There are representatives of the others, like H5 and H9 and et cetera, that have launched skirmishes into the human population, but have not established transmissibility human to human to make a pandemic. But that doesn't mean it won't happen. And
I suppose in honor of D-Day, we could say they haven't established a beachhead yet.
And were it to happen, given the extreme antigenic variation or differences, and the human population, if we encountered a different subtype, would be completely or quite naive to this. When we've talked before about what's the next pandemic, flu's on the top of my list.
Yeah.
I think flu.
Yep. And this paper just emphasizes that. It's incredible, the zoo of viruses out there in these creatures that fly around, right?
Yeah.
Mm-hmm.
Yeah, and this so one of the I guess main innovations in this paper is they point out this H9N2 clade, or is that a subclade? Before we get to the subclades, the H9N2s have themselves diverged a great deal within the birds and within the poultry, and they've seen this in the course of their, decades-long monitoring. And that's why one of the things they do in this paper is come up with a way to distinguish and categorize these subclades because they are different enough that we need to worry about them differently because they've got, and they're picking up, so it may be an H9N2, but it's picked up pieces from other, flu viruses, an H3N8 or something, or an H10N3, and incorporated those. So it presents as an H9N2, but it's got bits of recombination going on from elsewhere too.
And that antigenic variation in the hemagglutinin is, that's not in a general sense, that's not a new phenomenon. Okay.
No.
I taught this for years in the human viruses, antigenic drift within a subtype that can be sufficiently pronounced so that two subtypes that have drifted from each other but are still, for example, H1, can be almost not cross-reactive. All right, but they're still within the same hemagglutinin subtype. But this is a profound demonstration of this happening in birds, and as Alan said, a good way to categorize what's going on or catalog what's going on.
And because they're looking at poultry, these are domestic birds that are in close contact with humans at all times.
Yeah, and I should. All these samples are taken from. What do they call them? Live
LPMs,
poultry
live
markets.
poultry markets.
LPMs,
I was just about to say, I
markets.
forgot to say that. Every time I came across the abbreviation, I had to look it up again.
Yes.
So culturally, how widespread are live poultry markets? They're in New York City, right?
Yeah, we have them here.
Okay. So you can find
Yeah,
live
yeah,
poultry
yeah.
markets anywhere. Is that
Yeah, you can
fair
go
enough?
in. They're in Brooklyn. They're in the Bronx. You can go in and pick out a chick. I've been in them before. In Arthur Avenue up in the Bronx, they have them. Yeah.
The U.S. Post Office actually has specific regulations for mailing live poultry. You can mail poultry to people. Don't.
Okay,
I came
why
across
do you
this
know
somewhere.
that?
And it struck me as hilarious when I saw something about this. I was like, wait. Mailing poultry, and I looked it up, and yeah, the post office has regulations because this is something that's done mostly from farm to farm, but yeah, live poultry are there's a lot of poultry in motion, you might say.
Mm.
So in China, there is, in the Chinese Academy of Sciences, a Center for Influenza Research and Early Warning. They do monthly avian influenza virus surveillance across China. They've been doing this since 2014. And from this, they can have an idea of the diversity of avian influenza viruses in poultry, in LPMs, live poultry markets.
I was thinking as I was reading this too, because there are there's influenza surveillance that goes on globally, right? And there are networks of surveillance operations. But other than that, I don't know the detail.
There's the GISA, the Global Influenza Surveillance.
So this is one
like.
piece of the puzzle, but quite a piece, a detailed piece.
I think this is a fairly large piece. Yeah.
Right.
So from 2016 to 2019, interestingly, the positivity decreased, and the H9N2s became the dominant subtype in domestic poultry. Now, remember clade 2.3.4.4b?
Sure. Yeah.
Rolls right off the tongue.
Yeah.
H5N1 and H5N8. These are the ones who have spread globally and now in the U.S. or in cattle, right, and some humans. They've also had infections with lots of other avian subtypes like H3N8 and H10N3. These H3N8 and H10N3 have internal genes that are from H9N2. As you'll see, H9N2 is pretty dominant there, and it can grow in birds and in humans too. So it's got the right genes, the right stuff, and these often recombine and go into other subtypes like H3N8 and H10N3. So
your mention of internal segments prompts me to remind the audience that this is a flu has a segmented genome. So each segment, in effect, this is exaggerating, but in effect each segment has one gene. And if you have a mixed infection with two different viruses that have different gene segments, they can very easily reassort. Okay, so that's a big deal.
Yeah, the virus comes into the cell as 8 little pieces. Well, it's probably more than 8 little pieces, but you need 8 pieces to.
What was it? Five easy pieces? Eight easy pieces.
8 easy pieces. And if 2 viruses go into the cell, then they can just trade pieces.
So there's a concern about these H9N2s because they could give segments to other influenza viruses and make new ones, right? So they infect humans, obviously, but they're also widespread in poultry in not only Asian countries, but the Middle East. And as of July 4th, 2023, there were 125 human cases of H9N2 in 8 countries. And China has had the most, 112 cases. And this is now where we get into genotypes. They talk about the G57 genotype, which was dominant, and it has contributed these internal RNA segments to H7N9 and H3N8. So H9N2 is a concern. But 125 cases, it's not huge. So in this paper, they look at dynamics, they look at subtypes, host species in domestic poultry from live poultry markets between 2019 and 2023 in 10 provinces and municipalities in China. And they do a few other things as well. But they have lovely maps here which show you exactly, figure 1, if you can get the paper.
It is unfortunately paywalled.
They have all the provinces that they've sampled. It's 18,425 samples from 18 live poultry markets in 10 provinces between March 2019 and March 2023. 3,237 tested positive for avian influenza. So that's 17.57%. That's higher than May 2016 to February 2019, but lower than November 2014, 2016. Most of these were from chickens. 13,000 samples and 2,151 avian influenza virus positive isolates. And then ducks, 971 isolates from 3,400 samples, and the rest from the environment. Geese, pigeons. They don't have any cool bird names today like we had last time. They just got geese, pigeons, ducks. The highest positivity was in ducks, 28.48%, followed by chickens. So as you know, well, ducks can be, chickens are not wild animals, right? They're only farmed animals, but ducks can be farmed or wild animals. And they look at the distribution of the isolates across 10 provinces. In Shaanxi had the highest positivity rate, 34.92%. We look at the map. Where is that? That's the red one. It's all the way down in
Up in the
south.
north.
No,
Up in the
JX.
northeast. Well, it's all kind of toward the east of the country.
It's down by Taiwan, actually.
Yeah.
No, that's Fujian.
Yeah, Fujian, then Jiangxi is next to it, right?
Okay. No, Shanxi is up in the north. They have 2 pink on the map. They have 2 pink provinces.
Oh, Shanxi. What about Jiangxi? That's one.
Jiangxi is down in the south.
That's
the next one west from Fujian. So yeah, you go from Taiwan to Fujian, Jiangxi.
Yeah, that's the highest isolation rate, right?
Right.
And then they rank all the other ones. We don't have to go into that, but they have those data.
Is it fair to say that the waterfowl are the primary reservoir of flu? Or
yeah.
Okay.
Waterfowl. Yeah.
Right. So because one of the things I noticed, interestingly, is that the ducks have, you're probably getting here, the largest assortment.
Yes.
Of viruses.
Yes. Right. So that's what I was just getting to. The next bit is the ice. So they have 2 kinds of isolates. They have what they call pure isolates and impure. I think that's funny. So pure is just one HA or NA subtype. And then impure is more than one, which is, I guess, implying co-infection. So the pure isolates, they have 23 subtypes, and H9N2 is 85% basically, followed by a bunch of others. And the pure isolates were mainly gotten from chickens, 76%, and ducks, 21%. But in ducks, they had 19 influenza subtypes, including H9N2, a bunch of H5s, and others. There's so many out there. And 10 subtypes were in chickens. And again, H9N2 dominates in both chickens and ducks. So this is great because it's really a broad sampling of China in these live markets. And you get an idea of what's out there, right?
Yeah.
Here, when they're talking about, I was just noticing this, when they say 19 subtypes, they're calling subtypes that might have the same hemagglutinin but different neuraminidase, different subtypes. Okay, so H5 shows up a number of times. Well, I guess just twice in ducks with two different neuraminidases, etc. But man, what a zoo!
And then they have 473 impure isolates. This is a funny word. It sounds like they're passing judgment. And these are basically co-infections, and they document what these are. So again, H9 and N2 were predominant, particularly in chickens. So there were lots of combinations. We don't need to talk about that, but they're out there. So obviously co-infections happen, which is not surprising if you have a lot of look at all the viruses in ducks. And, ducks are social. They they hang out, and they infect each other, right? So they have multiple infections. What is it? That is Hickham's dictum, as Daniel says. You can have as many infections as you damn well please. So Occam's razor is, the simplest explanation.
Right.
But Hickham's dictum is you can have as many infections as you please. Because Hickam said Occam was not a physician. I am. All right. So they also, so all of these H9s belong to a lineage called BJ94. And so then they proposed a new nomenclature system for these BJ94 lineage viruses based on, the genome sequences, phylogenetic mapping, and so forth. And so they have this 3-level classification into clades, subclades, and sub-subclades. And they talk about what criteria they use, but they're basically using the genome sequence in order to do that, which they can obtain from obviously the samples. And so this gives you a framework for future isolations. You can put them into this framework. Now, antigenic variation. Of course, the hemagglutinin is the main antigenic glycoprotein on the surface, and antibodies against it can protect. They say against infection, but I would say mainly against disease, unless you have a lot of antibodies present early after infection. And so they wanted to see if there was any cross-reactive immunity between these prevalent H9 and 2 clades. So they make chicken antisera against representative strains in subclade 4.6. They were made, and they do cross-hemagglutination inhibition. So hemagglutination is when you take influenza virus and you mix it with red blood cells. And the viruses cause the red blood cells to clump because one virus will bind a couple of red blood cells. And so what they do in a little plastic well, they'll form a sheet coating the well. That's called hemagglutination. And you can quantify it. And if you put antibodies to the HA in there, you can inhibit it. So that's what hemagglutination inhibition is. So we have HA assays and HI assays.
So you're not measuring infectivity or anything. You're measuring antibodies binding to the hemagglutinin, basically.
That's right.
Binding to the flu HA.
The flu hemagglutinin,
right. Yes, exactly right. And so they look at antigenic distance between these clades, and they see a low level of cross-reactivity between the subclades, and that they say supports their classification of the H9 HAs. And they say this could help develop vaccines against H9N2 avian influenza virus.
Yeah, so you're taking antibodies against one subclade and using them against another subclade and seeing if you inhibit hemagglutination, right. And that tells you if there's cross-reactivity.
I'm looking at this phylogenetic tree that they created. And man, these H9 viruses just exploded during this period of time. And the diversity is striking. There's a lot of experimentation going on among these viruses with, probing their environment by changing their hemagglutinins.
It's like a Cambrian radiation of these viruses. They found a lucky spot and they're exploring it.
Lucky spot, yeah. And some oxygen present too. So, all right, so they look at reassortment. They develop a nomenclature system for the 6 internal genes and the neuraminidase, and they look at reassortment, and they see that among all the samples that they have, right? So they note that the genesis of the 5 human infected avian influenza subtypes, so that would be H7N9. We should remember this because you might encounter one day. H7N9, H10N3, H10N8, H3N8, and H5N6. They all got some internal genes from H9N2 avian influenza viruses. So that H9N2 is a great donor. Now, when they say human infecting, that's it. They don't really transmit effectively among humans. There are no outbreaks. I guess if there's a couple of cases, it's an outbreak, but it doesn't transmit extensively. So they're just sporadic spillovers from bird reservoirs. The ones that infect humans and transmit are the H1, the H2, and the N3. That's it so far. And, we're worried about H5, and obviously we're worried about H7 now. So.
And by the way, one good thing about a flu pandemic is at least we already have the naming system in place. We won't have to go alpha, beta, gamma, or this kind of thing.
Well, and there's this incredible history of technology for understanding and making vaccines and et cetera. So
the bad news, though, is that the vaccines we have kind of suck.
Yeah.
Back on the nomenclature thing, I was trying to find it, and I just have, where they talk about, because they've set up this system to be able to categorize these things, they have this 3-level numeric clade designation, but they may run out of ways to name things. So then they'll add a letter alias whenever they've run out of that. So, for example, a descendant clade of subclade 4.6.1 would be named A.1 instead of 4.6.1.1. If you follow that.
Oh, yeah.
Yes.
I got it all.
This is not for us. This is for the people who track this. And it's hard for non-science listeners, too. Both of these papers are very difficult. For me, too, because I can't remember all the abbreviations.
Yes.
And then they tell you what the abbreviation is here, and then on the next page they do something with it, and you can't remember. You have to go back and forth, and it's kind of annoying.
And all at the LPNs. LPMs.
Yeah.
LPN.
Live poultry markets.
Yes.
Live poultry market. Yeah, then there's LPG, liquid petroleum gas, remember?
Yes, right.
Okay, so the next thing they look at are amino acid changes that presumably help these viruses to become adapted to humans. They call them human adaptive mutations. That would be correct if it were in the gene, but they're always talking about like the Q226L mutation. It's an amino acid change, not a mutation. But I'm not going to argue with these guys that have.
You'll notice in my summary, I tried to call them amino acid mutations or changes, not changes. I said changes. Yeah.
So we already know a bunch of changes in influenza virus proteins that have an impact on reproduction in humans. So, for example, in the HA protein, the Q226L, glutamine to leucine, increases the binding of the hemagglutinin to human type sialic receptors. All right. So that allows infection of human cells and transmission among mammals. So obviously that's an important mutation, amino acid change to look at. The proportion of L226 well, now it's Q226L, so that would be the proportion of L226 has increased among H9N2 viruses worldwide. And so it's over 90% of available sequences since 2011. And from 2021 to 23, almost all, 99.46%, have this leucine at 226 in the HA. It's interesting.
Wow, if you
Very
look at this graph that looks at the proportion of that mutation going back to before 1995, the change is just dramatic. What is driving that? It's amazing.
A good question, because it's not in that many people, right?
No.
Right.
No, there's something else going on.
It's got to be doing something for the virus in the bird
birds?
host.
Yeah.
Or mammals,
exactly.
right?
Or there's a mammal
Or,
involved.
I suppose. But, and
there could be
there's the other one that you'll get to, I'm sure. NP undergoes a dramatic change as well. It makes me wonder if those are not, if they're in some synergy or something going on there.
Yeah.
Or just the
Very
patterns.
well
Because
be.
I don't know. But something's going on.
So then the next set of changes are in the RNA protein complex that is important for RNA synthesis. But they have a bunch of changes. K526R, for example, and a bunch of others. In the PB2 protein, this boosts replication in mammalian cells. Now, K526R, no, E627K, that was science can break your heart. Who was the guy on that episode? Kathy, you remember him. What was his name? Was it Madison, Wisconsin? Yeah, it was Madison, Wisconsin.
Oh,
ASV.
you're talking about Andy Maley.
Yeah.
Andrew Maley. He was one of he has worked on that. It just allows better RNA synthesis in mammalian cells. Some of these are rarely found in H9, but some of them, others have increased, including K526R and A558V, particularly after 2016. So the point here is that these known human adapting changes we have to keep an eye on, because if they're increasing, that means something is going on. And so they looked at the phylogenies of these viruses that lead to human infections, and they can see that some of these changes follow along the phylogenetic tree to viruses that ended up infecting humans, because they have isolates from humans, of course. And this is interesting. These changes have higher frequencies along branches that led to human traits than along branches connecting avian traits. So it must be something other than birds, right?
Yeah.
Because it's not in the branches. It's not in the branches. Okay. What else here? So the nucleoprotein, as Rich said, there are a number of amino acid changes that are here determinants of so there's an antiviral, a host cell antiviral protein called BTN3A3, very memorable. And these changes in the NP are contributing to evasion of that antiviral effect, and they say that could be important for zoonotic spillover. And another change in NP amino acid is involved in evading another protein, antiviral protein, the interferon-induced protein MXA. And so they actually looked at the sensitivity of H9N2s with some of these changes in them. And they find that H9N2 with one of these NP changes is at least partially resistant to MXA. So again, you have to keep an eye on these changes that are potentially involved in this case, evading immunity. Okay, receptor binding preference, right? This is really important because that's the first step in infection. And here's where they have a bunch of viruses here which have really long combinations of letters, and I can't remember them. But 4 H9N2 strains have HA, the L226, remember, which makes it bind really well to human sialic acids. And they have human receptor specificity. And they look at the infection of human bronchial epithelial cells, and they find that these the viruses with that change reproduce in those cells. And so they also replicate in chickens. So despite having this change, which allows it to bind human receptors, is also obviously binding avian receptors. They find some strange results. So they in A549 cells, some of these viruses don't reproduce, but they have the wrong change in the polymerase apparently, and that's the reason why they can't do that. So basically, you can look at the effects of some of these amino acid changes in reproduction in cells and in culture. So basically, if you have H9N2 with this leucine 226, PB2, K at 627, K or V, and then nuclear protein amino acid change, they can efficiently infect human cells. Okay, then they did some transmissibility studies in ferrets and guinea pigs. They have a figure here with a cage with ferrets and guinea pigs.
Yes.
And viruses shown not to scale.
No. A virus is the size of a ferret head.
They have experiments where they have co-housed animals or animals with either in a separate cage or with a mesh between them so that they can't touch each other. And so these 4 viruses that were previously used in the cell culture experiments, H9N2, they're looked at transmission in guinea pigs and ferrets. So the 3 H9N2s with HAQ226L could be transmitted between ferrets and between guinea pigs by contact. Okay. One of those failed. And remember, these have, let's see, WF 1998 failed. So what do I know about that? I got to scroll back. WF 1998. It has Q226. It doesn't have leucine, see? And so that's important at 226. So that one failed to transmit. You need to have good reproduction to transmit, right? You have to shed a lot of virus. Some of these viruses could transmit between ferrets and between guinea pigs. Virus respiratory droplets, which would be you put them in separate cages, and those have all the right amino acid changes in order to enable that. So those are the data. It's very cool that they're doing this surveillance. They have to do it. Other people shouldn't be going in and doing it. They can do it. They have the capabilities, obviously. Yeah.
So there are H9 viruses out there.
Yeah.
That can infect human cells in culture, even primary cells, and that can actively infect appropriate animal models and be transmitted.
Yep.
Even by aerosol. Watch out.
Yes.
So the interesting, they make a few statements in the discussion which are worth looking at. The first one is they say it remains unclear why circulation of H9N2 is sustained despite the availability of vaccines. You can't stop transmission with vaccines. And anyway, if they're in birds, how could you ever hope?
Well,
right?
I'm assuming they're talking about veterinary vaccines. Is that right?
Yeah.
Mm-hmm.
Yes, there are no approved human vaccines.
And they vaccinate
Chickens,
against these particular viruses, right?
H9N2.
So I guess that's
presumably in chickens and ducks.
Yeah. Well, maybe that's part of what's driving this, I wonder. Okay, depending on how vigorous the vaccination efforts are.
Yeah.
Yeah. Well, I think they vaccinate chickens, right?
Right.
But they don't vaccinate wild animals.
No.
So that it seems to me that, the chicken to chicken transmission is important, obviously, but if you have wild birds, they're going to be able to infect chickens at some point, right?
And also, a vaccine inducing sterilizing immunity would be pretty exceptional.
Right.
And
Yeah,
a flu vaccine in particular, human flu vaccines, if you get 60% efficacy in a season, you're doing great.
Yes.
So I'm assuming these veterinary vaccines are not radically better than that. So if they're Yeah, you vaccinated your whole flock, but they can still get it.
Yeah, so it seems to me that if you have a broadly distributed vaccine and it's it's far from 100% effective, that's applying a particular selection pressure on that flock.
Good point.
So the vaccines will prevent, they will prevent death, basically.
Yeah. Right.
But the chickens are still going to be infected. So I don't know what the criteria are. If you can't sell an infected chicken, right? So I don't know what happens in China. I know what happens here. I think here you can't. You got to kill them all. Okay.
But to what extent do we test?
If a flock here is positive, you kill everything.
Okay.
But if you don't test the flock?
Yeah.
Well, if you don't test it, then yeah. And so some farmers may not test it, right? So there's currently a big outbreak in some state. Daniel just mentioned it yesterday. And they may be killing 6 million chickens.
Wow.
You know how they kill chickens?
God, do I want to know?
I don't think...
Well, you can cut the head off. You can break the neck.
You can't cut the head off of 6 million chickens. It would take you 2 years.
Isn't that what a chicken processing plant does?
No, they don't do that for here. They have these big barns where the chickens are. They shut off the ventilation and they turn the heat up.
Ooh.
Oh.
And it takes a couple of days for them to die.
Ooh.
They basically cook them. And you can use CO2, but it's more expensive. And of course, for farmers, it's all about expense, right? Oh, my gosh. Anyway.
So, clearly, we're not culling all of the infected chickens in China because these all came from, what are they? LMPs? LPMs.
1 out of 6.
Live poultry markets.
1 out of 6 in the markets.
Yep.
Yeah.
Okay. So then they go on to say that maybe there's some immune escape going on, and that's why these vaccines don't work. That could be part of it, right?
And as Rich said, maybe the immune escape is because of the vaccine,
or at least some of the evolution, some of the drift. Like I was asking early on, what's driving this incredible burst of evolution in the H9 viruses? I guess you can't do the control.
No.
No, you can't. All right, human. So then they talk a little bit about human reports that, as you know, I've told you how many human infections. We have to keep an eye on these HA changes, the ribonuclear proteins as well. And, they have evidence in this paper for some of these amino acid changes allowing reproduction in human cells and also transmission in this ferret and guinea pig models. And let's see what else I wanted to tell you. So the H9N2 has donated a lot of internal genes to many other subtypes. And so this is a concern because the H9N2s are out there circulating and they can do that. They can continue. Okay, so then I'll repeat what Kathy said. So 3 factors are associated with zoonotic transmission of avian influenza viruses from birds to humans. 1, spillover opportunity. So you have to have the opportunity, like a live poultry market is an opportunity. 2, the capacity to be transmitted to and replicate in humans. So replication in humans is important. And then they say escape from human immunity. Because if it's cleared by immune response, it's not going to be able to transmit, right? So surveillance, man.
And then they go through and they say, check, check, check.
Yes.
Yeah.
All
amino
right.
acid changes lead you to each one of these.
And then this reminds me of the 1953 DNA structure article where at the end Watson and Crick says, it has not escaped our attention.
Escaped our notice.
Says, these results highlighted the non-negligible zoonotic risks of H9N2 avian influenza viruses. Okay.
Mm-hmm. One thing that was in the last part of the paragraph before was that they didn't find any H5N1 virus in poultry from their live poultry markets during their surveillance. I thought that was...
That's the one that's going around in cattle now?
Yep.
Wow.
Yep.
Okay.
Yeah, it's interesting. Okay, that's the easy paper for today.
It's the easy paper, yes.
The next one, we don't have a summary, but...
Actually, you could probably...
do the abstract.
Read, or they have
or do the in brief.
Yeah,
the in brief.
Did you do the authors, Alan, at all?
Yeah,
Maybe?
so I've got everybody highlighted here. We have 5 co-first authors. This co-first authors thing, it's like all the children are above average. But anyway, we have 5 co-first authors on this. So it's a paper, Cell Reports. This is open access. The article is Immune Imprinting Blunts Omicron Pathogenicity Fluctuations and Highlights the Essential Role of Cellular Immunity in SARS-CoV-2 Infection. Co-first authors are Zhaohui Wei, Hekai Yang, Jing Wu, Binbin Zhao, and Yameng Mu. And the senior author is Zhangning Liu. They are from Peking Union Medical College in Beijing, Capital Medical University in Beijing, and Wuhan Surface Biotechnology in Wuhan. So the in brief for this is Wei et al. Report the virulence of the Omicron variant of SARS-CoV-2 exhibits a fluctuating resurgence, while antibodies induced by prior immunization or infection may struggle to effectively neutralize the emerging variant. Memory T cells play a crucial role in mitigating its pathogenicity, which is a pretty good summary.
Yep.
There's also, as I said, it's open access, and there's a graphical abstract, although it is a rather complex graphical abstract that will probably take you 20 to 30 minutes to parse as you go through it and go look at all of the individual pieces. It does generally cover the highlights.
So I have a question kind of up front, but why did we have to go through the Greek alphabet, and then we got stuck at Omicron?
Yeah.
Does that mean that they just tired of Greek letters and then they just started giving these numbers?
It meant they were too fearful of my pie puns.
or is Omicron really just one thing with all these sub variants?
The argument I've heard is that Omicron, the Omicron subclades, if we may, are less different from each other than Omicron was from Gamma, etc.
That's always been my assumption.
That's been my understanding.
That these things are all.
Story. I do still think it was targeted at me.
That these things are all, these subvariants are all closely enough related to Omicron to still be sort of Omicron SIBs, as opposed to SIBs of one of the other Greek letters. Okay, that's been my assumption anyway.
What they're doing here is initially they look at the pathogenicity based on things like hospital admissions and lengths of stay to various waves from the initial alpha prototype wave, they call it, to the Omicron variants BA1, BF7, and XBB1, and they show that the Omicron variants are much less pathogenic than the original SARS-CoV-2, but they vary in their pathogenicity, and XBB1 in particular seems to be more pathogenic than earlier Omicrons.
But there's some caveats, that really modify that conclusion, but we'll get into that. Okay, just to remind everyone, SARS-CoV-2, the original isolate, rapidly changed, became alpha, then beta, then gamma, then delta, and then Omicron, which we are now backpedaling. We're stuck with Omicron. We're pedaling and making lots of subvariants, and these include BA1, BA2, BA3, BA4, 5, BQ1, BF7, XBB1, EG5, and JN1. Some of these will play into this paper, and you have to remember that.
Yeah, you have to remember all of these. Come on.
Yeah, there'll be tests.
So they say these subvariants are markedly less virulent than previous variants of concern, and they're more transmissible and enhanced capacity for immune escape. Now, personally, I find these statements unsupportable, in fact. So, as Daniel will say, Omicron is still killing people. So, the problem is, as you will see in this paper, if you look at what happens to people, that's an observational study or a real-world study, as they're called now. You say, okay, more people are dying at this time with this variant, but you don't know a lot of things about those people because it's a observational study where you're just taking people who are going into the hospital, and there may be other things that are influencing their disease. So in this paper, they go to an animal model.
Including the progress of the pandemic itself.
Yes.
So in the early, the first wave, the alpha variant, before we were naming variants, killed a whole lot of people, millions and millions of people around the world, as we all know. And those people who died in the first wave don't have the opportunity to die in any subsequent waves. So it's possible that if they'd been infected with Omicron, they also would have died. But since they're already gone, we don't know that.
Yes.
And then pretty much everybody in the world has been exposed to either the vaccine or the virus or both at this point, often multiple times. And so now we're dealing with a population that is no longer immunologically naive, and subsequent waves would be expected to be less virulent on that basis alone. There is some evidence Omicron is indeed less virulent, but as Vincent pointed out, people do still die of this.
And that immune landscape could show up as apparently looking different, not only in virulence but transmissibility.
Yes.
So these are tough words.
Yeah.
So, for example, they say these subvariants are less virulent. Then they go on to say, however, Omicron continues to result in fatalities. And so my point is, I think in people, observational studies are very hard to make conclusions from. Another point here that they make is between 2022 and 23, more than 80% of the Chinese population was infected with Omicron, BA5, BF7, and XBB1, despite widespread administration of 3 doses of inactivated vaccines. Well, it doesn't matter if they're infected. It depends whether they get severe disease and die or not, and they don't tell us. I don't care about infection. Like in the U.S., a lot of people are getting infected or were in this past season. The key is whether they get severe disease and die, require hospitalization, and so forth. So that's really what is important.
The other thing I had in mind all throughout this paper is the nature of the vaccines that we're talking about.
Yes.
They're talking about inactivated vaccines, which first of all, don't give a very robust T-cell response. And second, as I understand it, we're not nearly as efficacious as some of the vaccines used elsewhere, notably the messenger RNA vaccines, which were highly efficacious and induced not only a robust antibody response, but also a robust T-cell response. They talk about that in the paper, but it's important to keep that in mind.
Yeah. Yeah. Throughout this, they're talking about the inactivated vaccine.
And they also say all of this has led to a debate about the necessity of annual variant-specific boosters, especially for healthy people. And that's what we have here in the U.S.
What, the debate or the boosters? Both.
The debate and the boosters.
We have both. We have boosters and we have debate for sure. Right? Paul Offit is always talking about that. And, now the FDA is saying we want you to do clinical trials for boosters, blah, blah, all this stuff. So anyway, then the other part of this paper is a comparison of antibody-based immunity with T-cell-based immunity. And then, antibodies decline over time, but T-cell responses may be more durable. And they say there's evidence that T-cells can control and mitigate the infection, the severity of infection with COVID-19. And they're going to do experiments to do that. So they're basically going to compare virulence and immune evasion of these subvariants, BA1, BF7, and XBB1, following early vaccination and infection with previous strains, and they're going to look at antibody and cellular immune responses and the roles that they play for this, in this. Okay. So first of all, they retrospectively analyzed clinical manifestations of hospitalized patients. This is a big problem here. So hospitalized patients infected with the prototype, which was the original. Then we have BA1, BF7, and XBB1. Okay. And these patients, most of these patients had their first infection, and very few of them had vaccination. Okay, so we're assuming they're all immunologically naive. Now, they say most of the enrolled patients had typical clinical symptoms, but they say a shorter hospitalization duration was detected in patients with subvariants. So they point out themselves later that early in the pandemic, they kept people in the hospital longer for caution. We didn't know what to do with them. And then by the time we got to Omicron, we're kicking them out. So I don't know that duration of hospital stays has anything to do with biology, the virus, right? Oh my gosh.
I have a gripe with their symptomatic patients ratio graph too, where they're listing symptoms across the bottom and showing the proportion of patients that had those symptoms. My gripe is actually linguistic. They chose some ridiculously convoluted terms for some of these, like pharyngoxerosis, dry throat. I mean, just say dry throat if you mean dry throat. So anyway, that's a relatively minor complaint.
So they also pointed out that XBB-infected patients were older.
Yes.
So you can't compare anymore if you have to get everybody with the same characteristics, which you can do in a clinical trial, right? You can screen people and say this age, no comorbids, this whatever. But when you're doing an observational, you got a mix of everything, and so it's always confounded. But then, as Alan said, they look at signs and symptoms, right? So prototype strain, the original strain, had significantly lower leukocytes and neutrophil counts compared with people infected with Omicron. But lymphocytes had the opposite trend. I just want to know what's clinically relevant, right? All these measurements, what does it mean? Did the people die or not, right? What was their disease like? They look at coagulation dysfunction, inflammation, and then they see differences. Prototype strain and XBB had higher ICU admission rates compared with BA1 and BF7. The degree of lung damage caused by XBB and BF7 was greater than that in BA1. And then they say age factors blunt the conclusion that XBB is more pathogenic than BA1. I think this is all a mess.
The XBB patients skewed older.
I think this is all a mess, frankly.
It is confusing.
You can't really conclude anything from any of this.
But I think what they're trying to do here is use the data that they have in humans, because they're going to get to to cell and animal experiments later.
Yeah.
But they're trying really hard to find in the human data something that they can kind of assess. Well, and the human data are a mess because humans are a mess, and because of the course of the pandemic, the immunity changed, and some of these people are older than others, and so on and so forth. And so, I cut them a little slack because these are the data they can get.
It's nice that they tried to compile and report the data.
Yes.
Okay. Yes, that's fine. But it's just, as they say in the next section, to exclude the impact of interference factors like underlying disease, age, prior vaccination, infection, we're going to use animal models, which is not going to exclude the impact because they're not humans, right? I mean, you will get some information, and you can see the immunology is not bad, but
I would say of the human data, the one display that stands out to me is this one of the symptoms. Because there are several of these situations where XBB and BF7, not always in the same circumstances, but XBB and BF7 to some extent, BA1 stand out as having, being more symptomatic than the prototype. It's not clear cut at all, but there are some peaks that are distracting.
Yeah.
Yeah, I mean, I don't know what a lot of these differences mean, right? It's because they could be due to other things besides the virus. Anyway, so we're going to look at some animal studies now. They have my mouse models. They're going to look at BA1, BF7, XBB, JN1, and the prototype strain in humanized mice in a hamster model. So when you infect mice with BF7, I'm sorry I have to repeat all these because...
Oh, I'm glad you do.
BF7, XBB1, or JN1, they have significant weight loss compared with those infected with BA1. So this is the thing now that we have to remember also the order in which these arose. So BA1 was first, and then XBB, and then JN1. Okay? I think that's important because I don't know what would lead to these different outcomes in terms of evolution in a population.
Right. Well, I think it's also important because what we're going to get to here is XBB1 kind of consistently seems like the worst of the bunch. And I think it's important to point out it's not the most recent. So this is not some trend of the virus getting
no,
more
it's not a trend at all. No. So, for example, the prototype and XBB caused the most severe disease in mice. So the prototype is the original, and then XBB is kind of a few variants down, and then there's more after it. So that's weird, right?
It is weird. And the way they summarize it in brief is that the virulence of the variants fluctuates.
That's right.
Fluctuating resurgence. Okay, and that's the kind of the best you can do. It's descriptive, but it describes it.
Yes, and so this is in an animal model, right? So, they're using mice and hamsters, and it's just, I'm not sure what it means with respect to people. And I know it's a cop-out because I just told you that I didn't like the
you didn't like the people experiments either.
You don't like anything.
No, the thing is, the only thing you could do to really know is to challenge people, and you can't really do that. They challenged people with the original strain once, and that was it. But it's very difficult, and I think this variation makes it worse, right? Because it doesn't make sense in terms of evolution. Anyway, next they go to Syrian.
Well, it does make sense if what's selectively pressuring the virus is not related directly to the pathogenesis.
Well, look, in the end, the only thing that matters is fitness.
Yes.
In my
So
opinion,
the virus is
right?
trying to evade immunity. That's the big selective pressure on it.
Well, it's not trying, but it's selected for.
It's selecting.
Right?
It's being selected for evasion of immunity. I realized as soon as I said trying.
That's okay.
Yeah, ping me on that. And so the Omicron variant was highly successful. I mean, whatever we say about transmissibility, it really did, get everywhere. And one of the reasons that happened was probably because it was able to get past some degree of immunity. And then where do you go with that? You want to know why that happened, but does it affect pathogenesis? Well, not necessarily. I mean, it could be evading immunity and becoming less pathogenic as well, but coincidentally, it's just, changes in a confined system and things are going to affect other things.
It's very complicated because, well, of all the things we've said. And also, I think fitness drives all the evolution of these viruses, not increases or decreases in pathogenicity. I don't understand a decrease in pathogen. That would imply a lower fitness, and it doesn't make any sense to me.
I mean, is there any relationship between pathogenicity and fitness? I don't know.
Yes, I think so. I think so. Fitness is just making more virus in the end, right? And if you make more virus, maybe you're more pathogenic, but not necessarily.
Not necessarily.
Yes,
I'll give you an important link between pathogenesis and fitness. SARS-1 versus SARS-2. SARS-1, ridiculously pathogenic, right? Very deadly.
Right.
Didn't get around very well because the people who reached the contagious phase were hospitalized and on a ventilator. SARS-2 got around great because the people who were at the contagious phase were up and about and, maybe had a mild sniffle, but they felt fine to go out to choir practice. So the pathogenicity, the decrease in pathogenicity in SARS-2 actually increased its transmissibility, which is a huge advantage in fitness.
Well, yeah, it didn't increase the inherent transmissibility of the virus, but because of human behavior.
Because of human behavior, yes.
And I'm keeping in mind as all this, consistent or on top of what you're saying, Alan, or consistent with it, there's is something like 50% of SARS-CoV-2 infections were asymptomatic, right?
Yeah.
So while we're looking at symptoms and talking about pathogenicity and all that kind of stuff, half of the infections are going on unnoticed.
And those
Yeah,
infections are probably the ones that are driving a lot of the
Yes,
spread.
of course. The problem, Alan, is that SARS-1 and 2 didn't derive from each other.
No, they didn't derive from each other. But I'm just saying, fitness does not mean you have to make the host sick.
Yes, I'm just saying, but for a virus to become less fit is just weird. I don't understand that. Anyway, in hamsters, here the virus doesn't kill them. So they look at weight loss, and they find weight loss in BF7, XBB1, but not in BA1 infected animals. Again, this weird difference in virulence.
And BA1 was the
first one. And then XBB, where's BF7 fit in? I have to scroll back. BF7 comes just before XBB, and then JN1 comes last. Maybe we could remember that. With 4 PhDs, you think we could remember?
No way.
It's just letters.
It's just letters. That's right. There you go. So in terms of lung lesions, the prototype BF7 and XBB had more severe than did BA1. So the prototype, then comes BA1 and then XBB. But this up and down pattern still persists. Then they look at viral loads in the brain, which, I mean, they say there's evidence for viral infection of the brain, but I just don't believe any of that. So I don't think this matters at all, because I think this is a feature of an animal model that has nothing to do with humans. And they talk about having higher neurological risk in some strains than others, but I don't think that's a part of the COVID, basically. Yeah. So basically, in Syrian hamsters, prototype BF7 and XBB are more pathogenic than BA1. So got prototype, we got BA1, they're right next to each other. So the prototype is more pathogenic than the next virus. So that means it had to have gone down because BA1 followed the prototype. And then BF7 and XBB are more pathogenic. So
Fluctuates.
Fluctuation.
It fluctuates.
Oh my gosh. We're not even through the paper and it's already 4:20. Woo. Let's see if we can summarize this. Immune responses. They want to know if that has anything to do with what we see. So they looked at these whether they're cross-protective immune responses from prototype or BA1. They have prototype and BA1 inactivated vaccines, and they look at these in the Syrian, the hamster model. And so when you immunize hamsters with the prototype or BA1, they make neutralizing antibodies, which will neutralize the prototype or the BA1, depending. But cross-neutralizing is reduced for the other strains.
Not zero, but reduced.
Not zero, but it's reduced, right.
Well, because they're all still part of the Omicron family group.
That's
whatever.
Right. And so that's what you would expect. Both vaccines induced weak or no cross-neutralizing antibodies against BF7 or XBB1. But after challenge, still with lower antibodies, you still got kind of improved viral loads and gross lesions and so forth. Okay, so they say our study suggests that vaccination reduces the injury caused by subsequent variant infections, because the early vaccination, the prototype and BA1, which were early isolates, right? They can modify the disease caused by the subsequent variants. And this is a whole part of the debate about whether we should have variant-specific vaccines. They also, those are neutralization. Oh no, those are animal experiments. Let's move on to, I'm going to skip this part here. We're talking about neutralizing antibodies. So basically, they're going to find that reinfection broadens the cross-neutralizing landscape. So you get infected by, say, whatever strain is first, and then if you get infected with a different variant, it broadens the neutralization that happens with that strain. So they get serum at 21 days from primary infections with prototype BA1, BF7, or XBB. They have great neutralization against their own strains. Now, the prototype can induce cross-neutralizing antibodies against both BA1 and BF7, But BA1 only induces cross-neutralizing against the prototype strain. BF7 and XBB induce cross-neutralizing antibodies against the other 3 strains, and then reinfection boosts and broadens this neutralization response. Okay? And they don't really understand the mechanism of that. They don't even talk about it, actually. And they notice that, in fact, if you get reinfected with BF7, you get more cross-neutralizing antibodies against EG5 and XBB1. So something is going on with having a second infection or vaccination with the variant. Okay, what about the cellular immune response? So they go back to mice, and they have mice that don't make antibodies, okay? And then they look at protection against reinfection at 30 days after immunization. So in these antibody knockout mice, there's no antibodies detected at 30 days post-infection. But they have pretty good weight loss. They have pretty good lung disease. Not pretty good, but decreased lung disease, reduced viral load, alleviated histopathology, compared to mice that don't get anything, don't get any vaccine. So something else is going on there, right? These animals have no antibody, and they're not fully protected, but they still have some protection. We okay there? Everybody
Yeah.
okay?
Mm-hmm.
Yep.
No neutralizing antibodies. Okay. Then they say, okay, so there's some cellular immune responses happening here. So they do flow cytometry to look at lung resident and circulating antigen-specific memory B cells, antibody-secreting cells, CD4 and CD8 T cells before and after infection. And these go up in infected animals, and you get an increase in CD4 and CD8 cells in both the lung and in peripheral blood. And cytokine analysis confirms that those are the right cell types. So you get an increase in T cell memory after infection. And they look at different populations, and they find that the lung resident T cells are the most important for ameliorating disease, as you might expect. But there are also other kinds of T cells we won't get into, but they looked at all of those. And basically CD8 and CD4 cells in the peripheral blood were activated by central memory and other memory cells, where those in lung tissues were activated by other T cells.
So once again, T cells to the rescue.
Yeah, so T cells are important for, I would say, ameliorating disease in the absence of antibodies. They're both important, but T cells are interesting because they don't undergo the variation that the epitopes that are recognized by antibodies do. Right?
I figure T cells are saving my bacon here.
Probably, if you don't have any other issues. So then the last experiments, they're going to do adoptive transfer, where you take cell populations from an immunized animal and you put it into a non-immunized animal, and you ask what happens when you infect them. So they're going to take serum, which has antibodies in it, tissue-resident memory B cells, circulating memory B cells, circulating CD4 and CD8 T cells. They take all of those from mice at 30 days after infection, and then they transfer them into either the respiratory tract or the tail vein of uninfected mice, and then they challenge those with the infection, and they monitor body weight and lung histopathology. So the serum transfer improves symptoms. That's got antibodies in it, so that's no surprise. And memory B cell transfer did not reduce viral loads. Histopathological alleviation was found in the tracheal and lung tissues of the mice in the serum transfer group. So antibodies are doing pretty well. So the memory B cells alone didn't provide protection. So they looked at the role of T cells. They take the CD4 and the CD8 circulating cells, and they put them in the tail vein, and then they challenge them. And they give you nice improvement after infection. And basically, they think that the reason the memory B cells don't work is you don't have CD4 cells in there to help them. If you just give memory B cells, you're lacking the CD4s. And so they say basically CD4 and CD8 T cells play a role in infection, with the tissue-resident CD4s and circulating CD8s exhibiting more pronounced protective effects. So this further adds to the complication of Omicron and the variant-specific booster question, I think. So what can we conclude from this? Fluctuations in the pathogenicity challenges the prevailing view that variants universally exhibit attenuated variants compared to earlier strains. I would say that that's okay.
Yep.
I'm okay with that.
Yeah.
With that statement.
And that's
right.
That's an important statement.
Yes.
So then they say, as I mentioned before, in the early stages of the pandemic, hospital and discharge policies were more stringent. So you had long stays. So we can't really use hospital stay to confirm that, right? And then they say, external confounders like age, comorbidities, mixed infection, vaccination history should not be overlooked when interpreting the clinical outcomes. And I think many studies probably do that. Okay, fluctuations in pathogenicity, we don't know why, because it seems to not correlate with the order in which the viruses arose. I guess it doesn't have to, but it would be nice to know the mechanism, right? It's interesting that you have low cross protection against variants, but if you get a subsequent infection, that broadens that cross reactivity, which they don't understand how that arises as well. And we see the role of cellular immunity here in ameliorating disease. So I think one of the interesting aspects of this is this heterologous reinfection increases the breadth and depth of neutralizing antibodies after you've had your initial infection. So that could probably be used in some way in vaccine design, right? To try and make vaccines that give you broader, not just antibodies, but, antibodies. I think that's the issue that we have to address here. Okay, so that's Omicron. And I haven't touched an Omicron paper. We haven't in a long time. And we probably won't do one for a long time.
So what's the situation with boosters in the U.S.? The latest booster, is that to some recent variant? Is it a cocktail? I'm out of touch with this.
So Moderna just got an approval from FDA for, I forgot which variant it is. They did a very large clinical study. And so that's been approved. So you can, if you're over 65 or if you're under 65 and have comorbidities, you can get that booster, even if you've already been vaccinated, right? And if you've never been vaccinated, you want to get an initial course of vaccines, you would get that variant because it's the currently circulating variant. So that's been approved for Moderna, and that's the situation. Okay.
So that is a monovalent vaccine with a recent variant.
Yeah, I can tell you what it is because it was in Daniel's thing yesterday, which would be 12/24. Right here it is. Oh, the U.S. canceled its contract with Moderna for a bird flu vaccine. Did you know that?
Yeah, I knew that.
You know it because you're on top of these vaccine issues.
Well,
did you also know that Dr. Oz wants to bring bird flu infected ostriches from Canada to Florida and put them on his farm?
why?
What?
Because they want to kill them in Canada because they have avian influenza. But Dr. Oz, I guess, he said he wants to use them to study herd immunity.
Okay.
Yeah, they're a herd of ostriches, sure. Anyway,
I believe that would be a flock, wouldn't they?
a flock of orchids. I mean,
Flock
of
immunity.
ostriches.
flock
Immunity.
of ostriches. Yeah, orchids. All right, what am I looking for? I forgot now.
You're looking for
current,
the variant in the
Composition
latest,
of the current
yeah.
Moderna vaccine.
Let me search for. Oh, Moderna. There you go. That's the cancels contract. Well, we got a lot of Modernas here in this document. Okay, on May 30th, the FDA approved the COVID vaccine for Moderna, which is called MNX spike. The hell? What's the variant? They don't even tell you.
This is where we need the Brighton collaboration templates to tell you what's in all these things. It drives me crazy. It's probably too early for a packaging insert, right?
They call it a variant-specific vaccine. I cannot find what variant.
Okay, doesn't matter. I think that I have been immunized 7 times.
Wow.
And
that's a lot.
I've been infected twice, at least, that I know of. And none of this is recent. But I figure my T cells are in great shape. And I have a robust panel of antibodies.
Here you go. So it's a bivalent vaccine for BA4 and BA5.
Okay. Interesting.
Going way back.
Yeah, it's not even XBB, right? According to our paper, if I remember. BA4 and 5 are between BN1 and XBB, right?
Yeah.
But if it's a booster and you had, I mean, if God knows what you had first time around.
Probably the original.
Yeah, so that's
alpha,
in between.
Like alpha?
So it's you got BA1, BA4, 5, and XBB. So it's right in the middle there. It's not even a more recent one. But that's the problem with a big clinical trial. It takes time.
Sure.
Right? By then,
But if you got some other variant like the early alpha, and then you get this one, or you've been infected with a later variant, you probably have that breadth of immunity that we see in this paper.
Yeah, that's right.
And the T cell response, and you should be fine.
Yeah.
So I had the 3
or
original.
as fine as you're going to be.
I had the 3 original vaccines, which probably all of you did as well, right? And then I got COVID in December 23, so probably broadened my antibody response.
I have a very similar history myself,
Although, I did take Paxlovid, so I blunted the infection, so I don't know how much broadening I
was
got.
too late for Paxlovid by the time I realized what I had.
I had an infection recently that I tested once in the middle of it for COVID, and it was negative, and I decided that under the circumstances, that was good enough for me. And then as it was resolving, I realized I couldn't smell anything. Okay, so it's not only SARS-CoV-2 does that, but it is suspicious, right? I'd never experienced that before. That was pretty weird.
Yeah. Yeah, my smell went out for about a month and a half.
Ooh, long time.
It was annoying.
This was only a couple of weeks. Yeah, it is. I mean, mostly you don't notice it, but every now and then you think,
Yeah. No, mine went, I should say mine went out for a couple of weeks and then very slowly came back over about a month.
So we don't have time for letters, but I want to read just one because remember last time we had Brian who wants us to write letters to the states with the highest GDP. You guys were on, Kathy, you were on the last one.
Yes.
I wasn't there, but I'm aware of it.
Okay.
Yeah.
So we wanted to know what the states were because he said the top 7 states, the Magnificent 7, represent 38% of U.S. GDP, equivalent to the world's 3rd largest economy. So he drafted a letter that he suggests listeners send to the leaders of these states. And I wrote and I asked him, what are the states? So they are California, New York, Illinois, Pennsylvania, Massachusetts, Washington, and New Jersey. And Michigan is number 8. So it's not in the top, the Magnificent 7. But the whole point of this letter is that, 40% of NIH funding is being cut. So he suggests a 1.86% tax increase on incomes exceeding $1 million in these 7 states, which would generate revenues to backfill 38% of the impounded NIH funding. And he says there are over 400,000 people with 1 million plus incomes across the 7 states. I'm not one of them, that's for sure.
No, not me.
And so he thinks this would temporarily go, backfill the NIH budget, and then when it gets reversed, if ever, then they could get a refund, I guess. So anyway, folks, if you want to write, this is now, you can tell what states are here.
Right. And this from Brian has the whole letter again that he's proposing sending to the leadership in those 7 states. And it's an interesting proposal because those people who are going to get taxed would say, oh, I don't want to get taxed. So then they would potentially lobby for changes in the NIH funding.
Yeah.
Anyway, that will be in our sole letter this week. So if you want to do that.
Yeah. Thanks, Brian.
This tells you who can do it, right? Because you needed to know what state.
Yeah.
So thanks for sending that back. Okay. So let's do some picks of the week. Kathy, what do you have for us?
Well, I have something that's a paper, and it's from MSphere, which is the open access journal from American Society for Microbiology. And the title, it's called a perspective in virology. Virus species names have been standardized. Virus names remain unchanged. The first author is Murillo Zerbini, who's the head of the ICTV, and then a whole series of authors, including our friend Jens Kuhn. And the gist of it is that taxonomy is regulated by the ICTV, the International Committee on Taxonomy of Viruses, and taxon names are standardized so that people can recognize them and communicate and know they're talking about the right things. But a standard format for species names in viruses was lacking. And so there were the standards for orders, families, and genera when they end in virales, viridae, and virus. But there was no format for species names. And so in 2021, ICTV worked on this and adopted a standard binomial Linnaean format for virus species names. So it consists of the genus name followed by a, quote, free-form species epithet. And previously...
I think of an epithet.
I have epithets for some viruses.
Right.
Right.
Yes. Anyway, previously assigned virus species names that weren't compliant with the binomial format have been fully updated, and the names of viruses, or, quote, common names, such as yellow fever virus or human immunodeficiency virus, are not under the remit of the ICTV. They're not under its purview, and so, therefore, they've not been changed. So I think it's an interesting thing for... I was thinking about the Venn diagram of people who actually go and look at any of our picks of the week, intersecting with those people that are really interested in virus taxonomy, and how many are in the intersection of those two circles, I don't know, but I suspect there's at least a handful of them. And so this is for you.
Very nice.
Well, I think on TWiV we like to know this because we want to make sure we say the right names, right?
Mm-hmm.
Cool. That's great. Gosh, 24 authors. Here we go, 24. And Jens Kuhn is there. There you go.
Mm-hmm.
Okay.
Years ago, I had to actually write a little rap about taxonomy for Fields Virology.
You wrote the chapter in the Fields textbook.
Yeah, and it
right?
still exists. Actually, it was redrafted in the most recent draft, and Becky Dutch authored it and made me a co-author, which I thought was unnecessary. But I looked at the chapter, and they did actually. There's another co-author as well. I'm sorry, but I forget the name right now. But there is a lot of what I wrote that remains, one part of which is thinking about why do you need a taxonomy? It's all about you have to be able to talk about these things. You need names for them so that you can distinguish one from another, and so that you say a name and it means something to somebody and they know what you're talking about.
Imagine if the four of us called poliovirus something different.
Mm-hmm.
Right.
We wouldn't know what we're talking about, right?
And so actually, in order to talk accurately, you need a good taxonomy, okay, that actually means something. So all of this is worthwhile.
Yeah, if we can tell you something in the name, that would be useful, right?
Yeah.
Cool. Rich, what do you have for us?
Okay. This is something that another one of my bits that you didn't know you needed, okay? And you probably don't, but you might be entertained. This is an app that I use if I'm close to the water. That is open water, not lakes. And it's called the Real Tides and Currents Graph app, HD. It's also called Fly to Map. And I just have it's just an iOS app. Okay, it's not for Androids, so sorry. But it's very cool. Yeah. By the way, my mother, bless her deceased heart, was the only woman I've ever known who carried a tide table around with her. Okay? But this thing is your dynamic tide table. Anywhere you are, this you can punch a little button on this, and it'll home into where you are using GPS, and it will show you the tides, okay, in a dynamic fashion. Where you are in the tide cycle now, when's high tide, when's low tide, and then you can swipe along it, and it'll tell you, when the next high tide is and how high and how low it is. It's wonderful. Okay, it's in my folder on my phone called Marine that gets accessed when I go sailing.
Marine.
You really
Very
wanted
good.
this. Trust me.
It's good.
This is great.
Good nomenclature, Rich, for your folder. Alan, what do you have for us?
Well, inspired by Rich's pick and also yours that we'll hear in a moment, I decided it was time to go ahead and pick this, which coincidentally just came out. I had seen an earlier online version of it, but apparently it's in the current volume, the June issue of Current Opinion in Insect Science. Invasive termites and their growing global impact as major urban pests. The title doesn't really tell you what I thought was so cool about this, though. So invasive termites in the U.S. are a big deal, especially in the South. I'm sure Rich is at least somewhat familiar with this.
Sure.
There are these tropical termite species that have come into the country that wreak tremendous amounts of havoc and billions of dollars in damage. And it turns out, and this is an open access paper, that privately owned boats are a major, I guess, vector or fomite for transporting these into the U.S. and within areas in the U.S. And this paper has some nice images showing an active infestation on what looks like about a 30-foot cabin cruiser, infested boat with Capotermes gastroi, which I guess is an invasive one, and shows this is a fiberglass boat. But there's a lot of wood in a fiberglass boat. It's in often the core of the deck. It's in woodwork below decks. And the termites can get into it and infest it. And then they have an image, a graphic of a marina showing how those can spread from boat to boat within the marina.
Amazing.
And then as those boats go to other marinas, they take the termites with them.
Amazing.
So
Termites are awesome, right? I mean,
yeah, they're amazing
They can
creatures.
digest cellulose, right?
Yes.
And somehow
With a little
derive energy.
microbial help.
Yeah. So that's a good trick.
Yeah.
Hey, microbes, man, they're amazing.
Something that you said, maybe it was just, from privately owned boats.
Yeah.
What does the private have to do with
Well, it doesn't. That's
publicly
what
owned boats?
No, yeah. So this could happen to any boat.
Okay.
But the number of privately owned boats traveling around is, they're small boats, but there are a lot of them.
Got
moving
it.
around.
Okay.
Whereas, the Staten Island ferry, it only goes to two places.
Okay. So in a way, it'd be like private aviation versus
Airlines.
airlines
Yeah.
or something.
And
Okay.
privately owned boats are much harder to regulate because there are so many of them and they're scattered so many
Got it.
places.
Okay.
Especially, are they more likely to be wood, whereas the bigger boats are metal, right?
Yes, they're more likely, certainly most, I'm sure by numbers, the majority of wooden boats now are privately owned because commercial interests and states know that that's kind of a messy thing to get into.
Mm.
But
Beach.
even the fiberglass boats, as I say, have a lot of wood on them.
Yeah. It's my turn, right?
Yeah.
My pick is a video in Wired magazine So this was shown to me by Kim, our social media person here at Microbe TV, and it is it's put together by the American Museum of Natural History, and it's entomologist Dr. Jessica Ware answers insect questions, which is very good. I really like how she does it. And like, are cockroaches so resilient that they'd survive a nuclear war? Why do praying mantises behead their partners after mating? That one is very interesting. Why do crickets chirp? Are tarantulas dangerous? So it's fun, and she does a great job. So I like it because she's taking science and making it really appealing to the general public, right? It's just her there in a video, and she's got some. I looked at the mantis one, and she's got mantises crawling over her fingers, So, we can't do that. We can't have we do have viruses on us.
We have viruses all over us,
but we can't show you them.
Yeah.
I can't show you them. I can show you them on my shelf. That's about it.
She's got some really cool looking insects here.
Yes.
She's got all the I guess they're all cool. But I like walking sticks. I think walking sticks are awesome.
Oh, yeah.
And the mantises are very cool as well.
This is put together by Wired Magazine. They have a series here of different people, but I just looked at hers. Hers is good.
Yeah, I need to look at this. This goes on for a half an hour. Okay.
Yeah.
I got something to do. That's good.
This one,
there you go.
We have a listener pick, and Alan would be interested in this first one. This is from Jerry. First, I want to thank you for continuing to highlight the ongoing attacks on science and healthcare, as well as the consequences they will have on U.S. and around the world. On that theme, I would like to submit a listener pick. This is from a podcast that does media criticism. Their recent episode covers the long history of U.S. politicians and news media attacking supposedly frivolous science. Oh, gosh, remember the
Golden Fleece Awards.
Golden Fleece Awards.
Yep.
Mm-hmm.
For example, TWiP and sharp-eared clinical update listeners might remember the screwworm fly, spreader of a horrible parasite that eats living flesh and leaves enormous wounds on cattle and people before killing them. Back in the 1950s, a senator actually claimed that research into the sexual habits of the screwworm fly was wasteful. The podcast hosts note that the researcher from the paper ended up using that research to develop the program that has until recently eradicated the screwworm fly from North America, saving many lives and, less importantly, billions of dollars.
Edward Nippling.
Is
My grandfather was his supervisor at USDA.
Cool.
Yeah.
It's an interesting look at the history of anti-science and anti-intellectual attacks in the U.S. that might give younger folks context for our current situation. It also goes into the possible reasons behind these attacks. Thanks again, and keep up the good work. It gives a link for that. Thank you very much. That's great.
Nice.
Yeah, what was that? William Proxmire, right? The Golden Fleece Awards.
Mm-hmm.
Also, the lady who ran for vice president from Alaska, she was making fun of
Sarah Palin.
It's the elegans and fruit fly research, right? With no idea of how useful that's been. We also have a pick from Joy Rell, who writes, I am writing in with a listener pick. With all the airline crashes and issues with air traffic control, it seems like time to recommend my favorite series, Mayday, Air Disaster. As a Canadian, I'm proud that it's a Canadian production. The series combines recreations of airline disasters with the science behind how the causes were identified and the resulting changes made to improve safety. Interviews with the investigators or other professionals in the field, surviving pilots and passengers, are informative and give the viewer a personal look into the people behind the disasters. There is a YouTube channel featuring many of the episodes, and I have included links to my 3 favorite episodes, as well as a personal pick for Vincent. Features one of the first official investigations and has an old-time detective show feel to it, which I think he will like. The series really drives home the importance of independent investigations, investing the proper funds and training into air traffic control and so much more. My 3 favorite all feature incredible acts of piloting as well as very thorough explanations of the events causing each disaster. With everything being dismantled in your country, this series could serve as a reminder as to why the funding to science is so important. Links are listed below with season and episode number in case the links do not work. Thank you for all you do for science. Never stop fighting for knowledge. Joy Rell.
This looks links. Great.
Yeah.
Man, I'm definitely going to do this.
I'm familiar with some of these stories. The Gimli glider in particular is really, really cool.
Powerless Boeing falls from the sky.
Yeah.
Could it land?
Yeah, they landed at an airport. That was some spectacular piloting. They ran out of fuel.
Yeah.
Because they fueled up in Canada and somebody forgot to substitute. Somebody swapped gallons and liters. And they didn't have enough fuel to complete their trip, and they ended up gliding, and the pilot actually managed to extend the glide all the way to an airfield.
Wow.
Yeah.
Oh my gosh.
All right.
Thank you, Joy Rel. Thanks for those picks. This is TWiV 1225. You can find show notes at microbe.tv/TWiV. Send us your questions, your comments, your picks to twiv@microbe.tv, and consider supporting our work, spreading science to everyone. Go to microbe.tv/contribute. Kathy Spindler is a professor emerita, University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This was a lot of fun.
Alan Dove is at alandove.com, turbidplaque.com. Thank you, Alan.
Thank you. It's always a pleasure.
Rich Condit, Emeritus Professor, University of Florida, Gainesville, currently in Austin, Texas. Thank you, Rich.
Sure enough. Always a good time.
I'm Vincent Racaniello. You can find me at microbe.tv or on Blue Sky, microbetv.bsky.social. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.